Hello everyone, and welcome to this lecture. In this session, Dr. Erdogan will discuss the fundamental principles of bone biology and bone physiology. In the second part, which will be presented as a separate video, he will further elaborate on bone graft materials and the techniques utilized in oral and maxillofacial surgery.
In order to be successful in our treatments in implant dentistry, we must have thorough knowledge of physiology, metabolism, and biomechanics of bone tissue, because we will be directly dealing with bone tissue. And it is essential that our implant must integrate with bone, which is called osseointegration. This is a direct bondage between implant and bone tissue. This is one of the main tissues we're dealing with. That is why we must have good knowledge about bone. Although some patients have good bony tissue and nothing else is required for placing our implants, some patients are in need of bone augmentation, and that is why we must also have good knowledge in bone grafting materials. This is the objective. These are the objectives of this lecture. Bone, as you may remember, and maybe you know that it is a dynamic tissue. It's always in action, and bone always adapt to the loads under which is placed, and this law has been introduced by Dr. Wolff, and this is why called is Wolff's law. What does it mean? It means that if loading on a particular bone increases, the bone will remodel itself over time to become stronger to resist that sort of loading. This is called Wolff's law. So it means that if you apply forces on a particular bone, then the bone will remodel itself. It is dynamic tissue. It is not a stable tissue. This has been invented by Dr. Wolff. Just take a short look at the functions of bone. The first function is of bone tissue is to provide mechanical body support, motion, and locomotion. Locomotion is ability to move from one place to another. This is one of the main functions. Another function is that also concern us is supporting the teeth for biting and crushing food. Another function is supporting and protecting the critical organs such as brain and spinal cord. Another function is housing of bone marrow, which is the source of hematopoietic cells. This is very important, and is also responsible of calcium homeostasis. Of course, bone has many other functions, but these can be considered the main functions of bone tissue. Of course, it has many other functions. If you look at the composition of bone, it consists of cells and bone matrix. The cells that present in bone tissue are osteocytes, osteoblasts, osteoclasts, and bone lining cells. I will be talking about these cells in detail in later part of this lecture. We have also bone matrix, which consists of organic and inorganic part. Organic part establishes 35%, and inorganic part establishes 65% of bone matrix. In organic part, it is mainly composed of collagen, which is type 1 collagen, and it makes 90% of organic part of bone matrix. And other organic materials present in bone matrix are osteocalcin, osteonectin, proteoglycans, glycosaminoglycans, and some lipids, which are the ground substances. The inorganic part is mainly hydroxyapatite crystals. You can see the chemical structure of hydroxyapatite here in this slide. And mainly the inorganic part, the bone is inorganic, mainly the matrix is 65%, and it's mainly hydroxyapatite, and there are organic materials in it, such as collagen mainly, and we have cells deposited on these tissues, on this part, on this matrix. Now take a look at the important part of the bone tissue, which are the cells, bone cells. There are four types of cells in bone, which are osteoblasts, osteocytes, osteoclasts, and bone lining cells. Osteoblasts are derived from mesenchymal stem cells. They originate from mesenchymal stem cells. They are associated with the process of bone formation. They make bone tissue. They are responsible for synthesis of bone and matrix. Once the osteoblasts become embedded in bone matrix, I will also show a video about it, they transform to osteocytes. Okay, these osteocytes are less dynamic cells compared to osteoblasts. They are more stable cells. You will see them in the video later. And we also have cells that resorb bone matrix, which are called osteoclasts. And we have the last type of cells are bone lining cells. They are pretty much osteoblasts normally, but they are inactive. They reside on bone surface, and whenever the bone require osteoblasts for bone formation, they become osteoblasts. These cells are called bone lining cells. Now take a close look to these cells. Osteoblasts, which are, as I mentioned in the previous slide, originate from mesenchymal stem cells in bone marrow. Okay, the mesenchymal stem cells are undifferentiated cells in the bone marrow, so they can differentiate many types of cells, such as neurological cells or lipid cells or other type of cells. And one type of cells they can differentiate into is osteoblasts. So they differentiate, mesenchymal stem cells differentiate to osteoblasts. Their shape is large, cuboidal, and they cover all periosteal or endosteal surface where the bone is active. What they do is they secrete osteoid tissue toward the bone surface, and they make the bone matrix. Their shape show heterogeneity. They're not homogeneous everywhere. So their shape varies according to anatomical region. As I mentioned, they are responsible for synthesis, assembly, and mineralization of bone matrix. They make bone matrix, bone tissue. They form bone tissue. Osteocytes are originally osteoblasts, which are entrapped in bone matrix, and they differentiate in the osteocytes. So these all osteocytes are interconnected through tiny canicular systems. This canicular system provides diffusion of metabolites and signal molecules, and they combine the proceeding of bone metabolism. So they're responsible for metabolism of bone. They divide, even though it's slow, they divide with an average life of 25 years. And the spaces they reside is called lacuna. Bone lining cells, also called as osteoprogenitor cells or pre-osteoblasts, they belong to osteoblast family. They are inactive osteoblasts, and they cover periosteum and endosteum, the inner and outer part of bone. Their shape is flat. Unlike to osteoblasts, their shape is flat, and they have reduced cytoplasmic organelles, which indicates low activity of protein synthesis and cell metabolism. So when the bone requires osteoblasts, then some signal molecules are activated, and these bone lining cells become osteoblasts. And another important type of cells in bone biology is osteoclasts. unlike osteoblasts, they originate from monocyte, macrophage, hematopoietic cells. The originating tissue is different. Their function is degrading, eating the bone matrix. Okay, so osteoblasts make the bone, osteoclasts degrade the bone in various physiological and pathological conditions. Physiologically, they work; they are always active. The bone remodel itself all the time. That is, so when the bone is remodeling, osteoblasts make bone and osteoclasts resorb the bone. Compared to other bony cells, they have very large size. So in fact, you can see them under light microscope. Their size is between 30 to 100 micron. They are multinucleate cells, and it means that they have more than one nucleus. Then the number of nuclei could be between 3 to 30. Their cytoplasm is acidophilic because they must degrade the bone. That is why it is acidophilic, and it contains some organelles which are called vacuoles. Now take a look at the bone matrix. Bone matrix is synthesized by osteoblasts, and they are synthesized by the origin of bone matrix is some macromolecules, the osteoid tissue or pre-bone. It consists of water, minerals, collagen, and non-collagenous proteins. I already mentioned the ingredients of bone matrix previously. This osteoid tissue subsequently mineralize and form mature bone, and this bone matrix remodels throughout the life. Our body continuously remodels during whole life. It is called bone turnover, remodeling the bone, like forming and being degrading. It is called bone turnover, and it occurs in response to metabolic reactions or maybe in some pathological conditions. In physiological condition, the main purpose of the body is stable calcium level. Okay, so in our body we have calcium, and it must be stable. It must have certain levels. It could be certain number of calcium, percentage of calcium. If the calcium level is low, then in the plasma, then the bone, the calcium which is stored in bone must be released into plasma, and in order to be released, the bony matrix has to be degraded by osteoclasts. And this is maintained by hormonal and metabolic interaction. We know that about 0.7% of whole skeleton is resorbed daily and replaced with new healthy bone. And in every 142 days, turnover of the entire skeleton takes place, if you think about that rate, the ratio. The rate of bone turnover decreases with age and certain systemic factors, which means in older patients it is slower. In younger ages it is, of course, much faster. If you look at the cycle of bone remodeling, firstly, the bone must be remodeled, and pre-osteoclast, we have resting bone surface, and then osteoclast resorb the bone. It is not first, but it can. This is just showing one part of the remodeling cycle. In catabolic phase, the active osteoclasts come to the region. They resorb the bone, and the mononuclear cells are reversed, and pre-osteoblasts are activated, and osteoblasts come to the region, and the bone is formed. Then the osteocytes, the osteoblasts become osteocytes, and they entrap in the bone matrix. Now we have a video showing the bone remodeling. Here, gonna see the cells, what they do.
broken down and rebuilt in a process called bone remodeling.
This is bone remodeling. It's showing the bone remodeling
Bone
process.
tissue is broken down and resorbed by multinucleated cells known as osteoclasts.
These are osteoclasts
These cells are
resorbing
derived from monocytes, which originate
the bone.
Osteoclasts play an important role in liberating minerals and other molecules stored within the bone matrix.
These are osteoclasts resorbing bone.
Bone tissue serves as a repository for vital minerals, including calcium phosphate and various biologically active molecules, such as growth factors. The release of calcium from the bone can play a role in maintaining its homeostasis within the body. The cells responsible for building new bone tissue.
Now the osteoblasts are in action. The osteoblasts synthesize bone matrix.
Once active, they start to produce the organic component of bone, osteoid, which is predominantly made of collagen.
So the bony tissue is osteoid tissue.
Around the collagen scaffold to form hydroxyapatite, the major inorganic constituent of bone, which contains calcium phosphate. Bone mineral density, or BMD, can be used to estimate the strength of bone and to assess the risk of fracture. As osteoblasts form new bone tissue, many become
So after formation of bone matrix, they entrap in the bone matrix and they become osteocytes. And these osteocytes are interconnected with each other with caniculized system.
The structure, composition, and cellular processes that occur within bone allow it to simultaneously serve as a calcium reservoir, while providing structural support for the vital organs and for locomotion.
Now take a look at the macrostructure of bone. So far I talked about microstructure, microscopical situation, appearance of bone. Now take a look at the macrostructure. At macro, the bone is composed of cortical bone. Outside and inside it is trabecular bone. Histologically, the difference between two types, cortical bone and trabecular bone, are in the amount of solid substance and the geometrical layout of the bone matrix. So, in fact, the cell types or the bone matrix, whatever inside, is similar, but the difference between are the amount of them, the bone matrix amount, and the geometrical layout. The cortical bone is organized in bony cylinders around a central blood vessel, which is called Haversian system, and it provides mechanical support, while the trabecular bone is spongy, cancellous, which is softer, and bone that occupies substantial space within bone tissue, and it constitutes the medullary cavity of bone. And the medullary cavity is filled bone marrow, which can be the red or yellow marrow. Okay, and you can see here the photograph of cortical and trabecular bone here. So cortical bone is dense and its geometry is different, and the trabecular bone is more spongy compared to the cortical bone. And outside of bone tissue we have periosteum, and except the articular surfaces, the bone surfaces are covered with periosteum, which is a dense fibrous membrane. It is a membrane tissue, and the periosteum is composed of two layers, which is an outer fibrous layer and inside inner layer. The inner layer is also called the layer of cambium, layer. The outer layer provides toughness because it is dense and collagenous. It is composed of collagenous fibers and fibroblasts. It is rich in nerve fibers and blood supply. The inner layer is in direct contact with bone, and this is where the preosteoblasts reside. This is this area, this part of periosteum is a source of functional osteoblasts. This is this photograph, histological section, shows the inner part, the endosteum part of the bone tissue. Here you can see the bone, the periosteum, and the vascular supplies. As you see, in the periosteum is a thin layer which composed of cells, and inside the inner part it contains periosteoblasts. The endosteum is a place, this is a cavity, the medullary cavity, which is covered with endosteum. This endosteum consists of single layer of osteoblasts that form a very thin, delicate membrane. Architecturally, it is similar to the cambium layer of periosteum because of the presence of osteoprogenitor cells, osteoblasts, and osteoclasts. So you can, when you look at this photograph, you can imagine in a better way. This is showing the medullary cavity. We have the compact bone, and we have a Hauersian system. Outside we have periosteum, the inner layer and outer layer of periosteum. You can see it also has its vascular supply, which are the vessels that provides nutrition to the bone tissue. We have different type of bones at microscopic level, which are woven bone, composite bone, lamellar bone, and bundle bone. And the woven bone is also called as, maybe you can also call this osteoid tissue, which is the pre-bony tissue, which will go to maturization, at which will be lamellar bone in the later phase. This is called woven bone. We have a composite bone, which is a transitional stage between woven and lamellar bone. And we have phase 2, lamellar bone, which is adult or mature bone tissue. And in very few, limited areas, we have bundle bone. This type of bone is found around ligaments and joints. Firstly, take a look at woven bone. It plays a principal role during healing. This is a very quick formation of new bone in fractured bone or osteotomist segment. Initially, this woven bone is formed by osteoblasts, and it is turned to lamellar bone or mature bone in the later phases. Why it is called woven bone is because the fibrous matrix, which make this the matrix part of the main part of this the bone, this type of bone has woven appearance. It develops in disorganized fashion without lamellar architecture or Hauersian system. The organization of this type of bone is not very organized. It's very heterogenic. It has soft texture because of lower biomechanical strength, and it presents in embryonic bone tissue and pathologic bone tissue. We can also find this type of bone. And as I mentioned, as a result of maturation phase, it converts to lamellar bone eventually. Here you can differentiate between between woven and lamellar bone in the slide. The lamellar bone has organized architecture, show homogeneous status. The cells are dispersed in an organized fashion, but the woven bone is unorganized, as you see in the upper part. And when it gets maturation, become mature bone, it converts to lamellar bone and become more stiff, more tough, and more adult bone in the later phases. Lamellar bone describes mature, load-bearing bone of the adult skeleton. It forms slower than woven bone. It is well organized in its collagen structure and mineralized content. Its organization is better, and it is composed of multiple oriented layers. If you look at the types of bone formation, we have three types of bone formation. The bone formation, which are endochondral bone formation, intramembranous bone formation. In some bone pathologies, in rare circumstances, we have appositional bone formation. Endochondral bone formation occurs in long bones, maybe some vertebra or symphysis of the mandible. This type of bone is organized or formed by endochondral bone formation pathway. Here, as the name indicates, we have hyaline cartilage or chondroid tissue that will undergo to some process which eventually result in bone formation. So we have a hyaline cartilage tissue, and they're at the primary center of ossification. So we can consider this hyaline cartilage as a template, and at the primary center the ossification starts. The blood vessels invade to this avascular cartilage area, and this carry progenitor bone forming cells, and then eventually the bone is formed. Here you can see the formation of bone through endochondral bone formation pathway. We have hyaline cartilage, and then at the center the ossification starts. The vascular supply provides these cells which is required for bone formation, and eventually the bone is formed. In this, another totally different type of bone formation is intramembranous bone formation. The flat bones, such as skull, maxilla, and sternum, are formed through this type of bone formation, intramembranous bone formation. Here, the mechanism is in this type of mechanism, the bone grows in width. Here, the osteoblasts differentiate directly from pre-osteoblasts and lay down, and they seam osteoid tissue. Here we don't have any cartilage tissue involved. So here mesenchymal stem cells become osteoprogenitor cells and then osteoblasts. Osteoblasts surround themselves with matrix to become osteocytes. The matrix calcifies into trabecula with spaces, and they hold a red bone marrow. Eventually mesenchyme condenses and forms the periosteum at the bony surface, and then the superficial layers of spongy bone are replaced with compact bone tissue. Appositional bone formation is a new bone formation on the surface of a pre-existing bone layers. This is very rare type of bone formation, and it occurs in some bone pathologies such as Gares osteomyelitis. So here we have a bony surface already, bone established, and the new bone is formed as layers on this pre-existing bone tissue. This is called appositional bone formation. This is not a physiological type of bone formation. It is rather a pathological type of bone formation. And these so far were the bone formation types, but we also have bone formation in other types of circumstances, such as bone defect healing. Bone defects occur due to trauma or resection of bony tissue, or others maybe because of other type of etiology, and we have vanishing and lost compartments of bone, and the bone must restore itself through bone regeneration. And there could be physiological bone regeneration, which is continuous replacement of bone with matrix and bone cells. There is also reparative bone regeneration, which takes place when tissue is lost due to trauma and disease. And this bony tissue is reconstructed almost to its original architecture normally, but never 100% could be the same. And this repair process mimics the original bone formation phases. This bone regeneration can be activated by two type of bone formation, which is heterotrophic bone formation and orthotropic bone formation. Here we are in need of repair of bony lesion, such as socket healing, dental implant placement, or bone graft incorporation. In this kind of situation, there are activation of release of growth factors and some certain signaling molecules. And then after signaling of these molecules, this type of reparative bone formations occur. In heterotrophic bone formation, the osteoprogenital cells are far from the bone and they induce to convert the bone cells and they form new bone tissue. And the orthotopic bone formation, the osteoprogenital cells are found in close proximity onto the existing bone, such as they present in periosteum, endosteum, and intracortical canals. So far, I talked about the main principles of bone, histology, bone physiology, and microscopic and macro structure of bone tissue. And from this part on, I will continue in a particular part that concerns us more in implant treatment, which are the structure of alveolar bone, maxillofacial bone, and I will continue with the bone grafting materials in the later parts. And it will be in the second part of this lecture, in the second part of this recording. I'll see you in the second part of the lecture shortly.